Hey everyone, welcome to this explainer. We are jumping right in today with an absolutely mind-blowing journey. We're going to travel all the way from the extreme environments of early Earth, where the very spark of life began, right down into the microscopic, bustling factories that are working away inside your own body right this second. So think about it. Where does a cell actually come from? It's kind of the ultimate mystery, isn't it? We look around at plants, animals, each other, but to get to the bottom of this, we actually have to rewind the clock roughly 3.5 billion years, taking us straight to the boiling hot springs of our very primitive planet. To figure this mystery out, here is our game plan for today. Origins in the hot springs, seeing the invisible world, the tiny living factory, boundaries and osmosis, how cells multiply, and finally, the continuity of life. All right, let's kick things off with part 1, origins in the hot springs 3.5 billion years ago. Picture the extreme, totally hostile environment of early Earth. Researchers actually think life might not have started in the calm, open ocean, but in some seriously harsh, changing spots, like boiling hot springs. Take the Puga Valley in Ladakh, for example. The water there literally reaches the boiling point, but these crazy heat-loving bacteria, called thermophiles, just absolutely thrive in it. And here's the really wild part. Scientists studying these springs noticed calcium carbonate forming rapidly all around them. That deposit might have shielded early organic molecules from intense radiation, basically acting as the very first protective membrane. And that membrane, that is exactly what officially defines a living cell. Moving right along to part 2, seeing the invisible world, the technological leap. Okay, so if cells are the fundamental building blocks of everything, how on earth did we even discover them if they're completely invisible to us? Well, it all comes down to tech. Think about the number 0.1 millimeters. That is the absolute limit of the human eye's resolution. If you throw two dots closer together than 0.1 millimeters, your brain just kind of blurs them into one single dot. Since almost all cells are way, way smaller than that, we absolutely needed a massive technological leap to understand the world beyond our naked eye. Back in 1665, Robert Hooke used an early light microscope, which could magnify things maybe 200 to 300 times, to look at a piece of cork. He saw these tiny box-like compartments and coined the term cells. Fast forward to today, and we've got the modern electron microscope. Instead of regular visible light, this incredible machine blasts a beam of electrons to reveal fine structures at the nanometer scale. Just to put that into perspective, a nanometer is 1 billionth of a meter. That is the game-changing technology that finally let us see exactly how a cell does its job. Which brings us to part 3, the tiny living factory, structural and functional units. Now that we can actually see these things, let's step inside the cell itself. Think of it like a highly coordinated, microscopic living factory. But, not all factories are built the same. First, you've got prokaryotic cells, like primitive bacteria. These guys are tiny, just 1 to 10 micrometers, and they don't even have a well-defined nucleus. Then you step up to eukaryotic cells. These are the larger, way more complex cells that make up plants, animals, and, well, you. They range from 10 to 100 micrometers and feature a membrane-bound nucleus, along with a bunch of other specialized compartments. If this eukaryotic cell is a factory, the nucleus is absolutely the main command center. It holds your chromosomes, which carry all your inherited DNA, literally the master blueprint for your entire body. But get this, here's a crazy, surprising fact. Mature human red blood cells, they actually kick their nucleus out. By getting rid of their command center, they free up physical space to pack in way more hemoglobin, which means they can carry a lot more oxygen through your bloodstream. The trade-off, though, is that without a nucleus, they can't repair themselves, so they only live for about 120 days. All right, part 4, boundaries and osmosis, protecting the factory. So let's head to the outer edges of our factory. How does a cell control what comes in and what goes out? It's all about the cell membrane, and it is unimaginably thin, just 7 to 10 nanometers thick. Scientists explain its structure using something called the fluid mosaic model. Picture a lipid bilayer. So 2 layers of fat molecules with various proteins just embedded right inside them. And it's not some stiff, rigid wall. It is totally fluid. Those molecules are constantly moving sideways, flipping and rotating around. It acts as a highly selective security guard, letting certain substances pass through while totally blocking others. This membrane has to constantly deal with different environments through a process called osmosis. If a cell hangs out in an isotonic solution, meaning the concentration of solutes is perfectly equal on the inside and the outside, everything's cool, no change. But if you drop it into a hypotonic solution, like pure plain water, water rushes into the cell and it swells up like a water balloon. On the flip side, in a hypertonic solution like super heavy salt water, water rushes out and the cell shrinks. That is exactly why if you leave a piece of potato in a 20% salt solution, it'll visibly shrivel up after just an hour. Now, plant cells have a really brilliant workaround for this. Since a plant can't just up and walk away when the weather gets bad, they have an extra super rigid covering outside their membrane called a cell wall. So if a plant cell takes on a massive amount of water, it won't pop. That rigid wall holds the whole structure together and maintains its shape. It's the exact reason plant roots can just soak up vast amounts of water from the soil without literally destroying themselves. Okay, let's shift gears to part 5, how cells multiply, growth, and repair. Here's a big question. If a cell can only grow so big before it gets inefficient, how do organisms actually get bigger, or how does a scraped knee heal? The answer is cell division. Cells don't just infinitely expand, they multiply. The most common way they do this is through mitosis. Basically, one parent cell copies its DNA, splits right down the middle, and produces 2 genetically identical daughter cells. And this isn't rare. Every single day, an estimated hundreds of billions of cells in your body are replaced using this exact process. It is the absolute engine keeping you going, growing, and healing. But there's another way cells divide, called meiosis, and this one is a real game changer. This only happens in reproductive organs to create gametes, like sperm and egg cells. Instead of just making exact clones, the parent cell actually goes through 2 divisions. This incredible process halves the number of chromosomes and totally scrambles the genetic information. So what does that mean? It means children will look like their parents, but they are never exact copies. It's the biological driver behind all human diversity. Of course, all this dividing has to be kept on a very tight leash. In animal cells, there's this crucial mechanism called contact inhibition. When growing cells finally touch their neighbors, they naturally just stop dividing. It's an automatic, built-in brake pedal. But if that genetic control system breaks down, the cells lose that contact inhibition. They just keep multiplying uncontrollably, piling up and forming tumors. And sadly, that is the fundamental root of how cancer cells operate. Now, as crazy as it sounds, the exact opposite of controlled growth, controlled death, is just as important. Cells actually have a genetically hardwired self-destruct sequence known as programmed cell death, or PCD. And honestly, you should be pretty grateful for it. When you were just a tiny developing embryo in the womb, PCD specifically targeted and eliminated the cells growing between your fingers and toes. Without that intentional, organized cellular self-destruction, every single one of us would literally be born with webbed hands. How wild is that? Finally, we arrive at part 6, the continuity of life, cell theory. Let's zoom all the way back out for a second. How did the scientific community actually piece all this microscopic madness together? Well, it happened step by step as our technology got better. Like we mentioned, Hooke spotted the first quark cells in 1665. But it took until 1838 for Matthias Schleiden to figure out that all plants are made of cells. Just a year later, Theodor Schwann proved the exact same thing for animals. And then, in 1855, Rudolf Virchow dropped the final critical piece of the puzzle, completing what we now call the classical cell theory. This work was absolutely revolutionary. It totally unified the study of biology. Think about it. Whether you're talking about a microscopic bacterium, a massive towering redwood tree, or you, sitting there watching this explainer right now, we are all, every single one of us, products of these tiny, hardworking, incredibly complex living factories. And it all rests on Virchow's mic drop statement, all cells arise from pre-existing cells. Just let the weight of that sink in for a second. It means there is a literal, unbroken chain of life. Cells continuously dividing, multiplying, and passing down the blueprints, stretching all the way back to those boiling hot springs 3.5 billion years ago. We are completely connected to the very origins of life. But what happens next? The source text points to this jaw-dropping experiment where scientists literally scooped the DNA out of a bacterium and replaced it with entirely synthetic DNA. And it worked. The synthetic DNA took over and ran the living cell. If we are figuring out how to program the very code of life from scratch, what does the future of synthetic cells look like? And what kind of massive ethical questions are we going to have to face when we start building these biological factories ourselves? It is definitely something to ponder. Thanks so much for joining me on this incredible journey to the center of the cell, and I'll catch you next time.